Cool. Hello, everyone. My name's Dr. Molly Ann Williams, and I am a research scientist and product developer at Naturemetrics, who are based at the Surrey Research Park. So today I'm going to talk to you a bit about how we are using eDNA technology to tackle the biodiversity crisis that we are currently hearing so much about on the media and in the news. So who are Naturemetrics and what do we do? Well, Naturemetrics are the world's leading eDNA provider, and don't worry, I will get back to what eDNA is. I might use that word a lot, but I will explain, so don't worry about that. We are working with academics, governments, NGOs, and businesses all across the globe to help, well, in the pursuit of this nature-positive impact that we hear about, to develop nature-positive actions and activities within these businesses and NGOs that we are working with. This ranges from the water sector to oil and gas to conservation. So we work across a huge range of sectors, and it all comes down to protecting the environment and doing this through a non-destructive means. So biodiversity, I think we hear a lot about. I think a lot of people don't really know what it is. It's kind of talked about a lot, and it's become a buzzword. But when we think of biodiversity, we consider kind of three main factors. And I often compare this to pizza, which sounds a bit bizarre, but hopefully you'll get my gist as I go along. So the first thing we consider is variety. We want to have lots of different species within an ecosystem, just like you want lots of different types of pizza. Our second is quantity. No one just wants one slice of pizza. We want to have lots of it. So we want a huge variety of species, but a high quantity of that as well. And our third point is quality. So it's no good having lots of one type of species or even a few types. We need lots of different species who work together to have really good ecosystem services that help save the planet. So how do we normally monitor this then? Well, traditional methods for monitoring biodiversity can be highly time-consuming, highly expensive. They involve sending out big teams into the field to physically see and capture the organisms that we're interested in. And this isn't always possible. We might be working out in the middle of the rainforest or at a mining site, even some of our clients, and you can't send these big teams into these sites to look. They're also can be fairly destructive. If you're trying to capture the species, so in the case of fishing, you have to do something called electrofishing or trapping, and this could actually harm the organisms that you're trying to protect. And that's where Naturemetrics come in. So we are using something called environmental DNA. And this is effectively genetic material that we can extract from environmental samples, be this water, soil, sediment, or even air, to track the organisms that are present in an environment. So you might know humans, probably all over your house, it gets a bit dusty. We're constantly shedding DNA into our environment. And organisms are doing the same thing. So this goes from bacteria, the tiny little things, all the way up to humpback whales or massive mammals. And we can track all of this just using DNA. So our kits are fairly simple. We're actually demonstrating them out in the sun if you want to come and have a play with a paddling pool. But we can simply take a water sample. So this is, as I said, really non-destructive. Take a water sample, pump it through a filter using a syringe, and then at the moment, that filter is just sent back to Naturemetrics here in Surrey. We can then look at this for lots of different things, and we do two main approaches. Our first one is what we call meta-barcoding. Buzzword, sorry. Our second one is what we call single species analysis. And our meta-barcoding approach is effectively a whole picture. So this is a snapshot of everything that's going on in the environment. So this is that kind of big scale, what is in our environment altogether. So as I said, from really big creatures down to bacteria and algae. Our second approach then is our single species, and this uses something called qPCR, which I'm sure, because of the dreaded pandemic, you are all as well versed in as I am. We hear loads about PCR, but actually all it is a DNA detector. So in the COVID tests that we've been having, they're effectively looking for a COVID virus, and we can do the exact same in the environment. So you might have, say, invasive species like signal crayfish, or you might have protected species like white claw crayfish, and you might be trying to see if you have them in your environment. And this is really important when we look at conservation, for example, with bat detection. If you want to do a loft conversion, you might have people come in to see if you have bats living in your loft. You can't just go and convert it. And we can detect all of this just using eDNA, so you don't need to see them at all, which is fantastic. The problem is then, the current way we do it, which is great for me as a scientist, but not so good for areas where they can't have custom-built labs like we have here, is that it's fairly costly still, and you need trained experts to carry out this analysis. So we're looking at harnessing new technologies to move away from lab-based detection. And this is where I'm going to talk about something called CRISPR-Cas technology. So has anyone heard of CRISPR? Yeah? A few of you? Cool. So you might have heard of CRISPR because it won the Nobel Prize in Chemistry back in 2020. And it's commonly associated with the medical field. So we've heard of CRISPR babies being a new thing for genome editing. It's, again, came about because of COVID a lot, because of diagnostics. But this is all for kind of point-of-care testing, purely based in the medical space. And it's currently really not used for the environment. But there's no reason not to. CRISPR is just a DNA detector. If we can test for pathogens on site in a hospital really quickly, there's no reason we can't take this out into the field. And this can be really, really important when we go to areas like the Global South, where they don't have the resources to set up these big labs. And what we want to do is we want to provide kits to places like this so they can go out, they can look for invasive species that need to be detected rapidly and on-site and start building in their eradication programs to get rid of them before they become a problem. We've actually developed some work already in this field, and one of our research scientists was out in the Ivory Coast doing in-field testing in a really kind of cheap lab. So they did set one up, but it was very much portable equipment that could be done just at the side of the field if you needed to. And we're hopefully developing this further using, as I said, this CRISPR method. So that's kind of our overall vision then, I guess, is where we see this going and using these new technologies, is that ultimately we'll get rid of scientists in the lab, although hopefully not too much because I'd like my job, and bring this towards citizens being able to do this. And 10-year-olds can go out and take samples and they can give that data back to us. And it's actually really fitting, following on from the talk you just did, and getting more people involved in STEM, especially if they haven't really heard about it, and all these kind of different jobs. Like I know my field is quite a niche thing and you might not necessarily know about it, but if we can get this away from scientists in white lab coats and take it back into the field and encourage more people to do this themselves, then that's kind of the route we see this going. And we really hope to get people on board with it to bring it not only to low resource settings, but also here in the UK.